Great. Thank you very much. Sorry, had to find the unmute button. Well, it's very nice to be here and see you all and get a chance to connect. I'm excited today to share some of our ongoing efforts to understand the genomic mechanisms that allow populations to adapt to local environmental conditions in the oceans. And a lot of the work, or most of the work I'll be sharing today actually was led by Maria Gobian, who recently finished her PhD in my lab. But it also draws heavily on work by three former postdocs, Arne Jacobs, Aaron Wilder, and Anna Tigano, and then our long-term collaborator on the silverside work, Hannes Baumann at the University of Connecticut. So I realize this is a very unoriginal way to start an evolution-focused talk, but what I wanted to draw our attention to was thinking about how there's such an amazing diversification among organisms that's very obvious to us just by looking at the world around us with our naked eye. And that level of biodiversity is, of course, extremely important, but it's also becoming more and more recognized that there's another kind of biodiversity and diversification that ecologically can be really important, but that's much less obvious to us, that which exists within species and can be much more cryptic. So it's easy to understand when a species is distributed across a big mountain range or a very fragmented habitat. It's easy to think about how diversification happens over time. But what about in more open environments like the oceans? So traditionally, it was believed that these kind of open environments like oceans that have few obvious barriers to gene flow, that we'd really see genetic homogeneity within species over very vast spatial scales, because there'd just be too much gene flow and connectivity to allow any adaptation to local environmental conditions. But we now know that, sorry about that, that's not true, and that local adaptation is actually much more widespread, even in marine organisms, than we used to believe just a few decades ago. And one of the species that has really helped change or has really contributed to our changed perception about this is this little unassuming looking fish here, the Atlantic silverside. So these are small estuarine fish, get about this big, and they're distributed all along the east coast of North America. And this is actually one of the steepest thermal gradients, marine thermal gradients in the world. And being ectotherms like fish, you might expect them to grow then much faster down in these toasty warm water in the south than they would up in the cold waters in the north. But when we go out and sample silversides in the wild, we see that they reach more or less the same adult size during their 1 year lifespan across all latitudes. It's a very different picture when we take them into the lab. So these are fish here, silversides, that we've reared under common conditions in the lab. They're exactly the same age, the only difference being that these top fish have parents from New York and the bottom fish have parents from Georgia. So you can see that there's a huge difference in the intrinsic growth capacity in these different populations. And this was first discovered by David Conover, who was a professor at Stony Brook University, and ended up dedicating a lot of his career to studying local adaptation patterns in Atlantic silversides. And he demonstrated with common garden studies that there's a very tight correlation between the intrinsic growth rate or the growth capacity and the length of the growing season across latitudes in silversides. And with both these common garden studies and a whole host of field studies, his work and the work of his group have characterized really well the selective drivers behind this pattern, showing that in the north, when the growth conditions are poor, the fish need to attain a certain size to survive the harsh winter. So there up in the north, there's been very strong selection for faster growth. But then there are trade-offs with predator avoidance that then has caused a decline in the intensity of that selection for faster growth. So there's been selection for slower growth in the south. And this is a very interesting type of genetic variation because it's then counteracting the genetic influence on the trait basically counteracts the environmental influence. So the colder temperatures in the north would make fish go slower, but then they compensate for that genetically, so that the genetic influence on the trait is to grow faster, and then vice versa in the south. So that means that this type of variation is called counter-gradient variation. Genetic influence on the trait opposes the environmental influence, which is a really interesting type of variation, because it then means that there's less phenotypic variation in the wild, and that kind of adaptation really becomes cryptic to field sampling. It's only if we're able to isolate genetic and environmental induced components on trait variation that we can even detect that there is this adaptation going on here. And it's not just in silversides, it's not just in growth rates, but it's a whole host of other traits that show either counter-gradient or co-gradient variation. So some related to growth, like lipid energy reserve, metabolism, rates of energy consumption, but also fecundity related to egg, and behavioral traits like willingness to forage, and even their sex determination system also shows clinal variation. And what's most remarkable is that we know that the genetic variation in all these traits is maintained even over pretty fine spatial scales of a few hundred kilometers, even though we know both from earlier mitochondrial DNA work and from microchemical tracing that there actually is quite extensive gene flow up and down the coast here. What we also know is that growth rate and other traits can evolve really rapidly in the species. The reason why I first got working with these guys was actually to follow up on an experiment that was published back in 2002 that showed that size differences shown in this picture here had evolved over just 4 generations of strong experimental size selection. So I'll come back to that a bit later, but the point here being that we know that silversides seem to show this remarkable capacity at rapid adaptation and adaptation to local conditions despite gene flow. And these patterns have been really well studied at the phenotypic level. So we have a host of context information on the phenotypic patterns, but only quite recently have we been able to look at the genomic basis for this in silversides. That's a complete non-model organism where we had to start from scratch with developing the genomic resources. So in my lab, we've been really interested in looking into what is the genomic basis for this amazing adaptability in silversides, and in particular, the geographic adaptive climb across latitudes. And is there something about the underlying genomic architecture that helps the species maintain local adaptation, even though there is a lot of gene flow? So basically what we're testing is this theoretical prediction that when gene flow is strong, local adaptation should proceed primarily through mutations of large effect or tightly linked clusters of smaller effect loci, because any individual loci that only have very small effects on traits would just be any selection on such loci would just be completely swamped out by gene flow. So we used kind of a two-pronged approach to get at this. We started out by looking at just sequence data, population genomic data, to look at geographic patterns of variation and identify where in the genome do we see patterns of selection across latitude. And then we coupled that with a more top-down approach where we started looking at those traits that we know to show local adaptation. And then we mapped those traits with QTL mapping to the genome to see where in the genome are those traits controlled. And then we compared the insights that we gained from those two approaches. So if we start with the bottom up, looking at population genomic data, we generated low coverage whole genome sequencing data, which is an approach we've also been very excited about in my lab, of trying to make whole genome analysis very cost effective so that we can look at large sample sizes across many different locations. And in this study here, we're looking at 12 locations with 50 individuals per population sequenced to just about 1.3x sequencing depth. So obviously too low to call individual genotypes. So all the results I'm going to be showing you are based on analysis within a probabilistic framework that takes genotype uncertainty into account in the downstream inference. So if we start out by just looking at the overall population structure, here we see just looking at a PCA where the dots here, each dot is an individual fish colored by its sampling location shown on the map over here. And we clearly see that there's very little differentiation. Basically the populations in the southern half of the range over here all fall. There's a little bit, looks to be a little bit of isolation by distance, but they all fall in a tight cluster here, where the more northern populations separate out more. And this translates to effective migration surfaces that suggest very high gene flow here in the southern part of the range, and more restricted gene flow with clear barriers in the north. So if we are going to now look, this is just the overall genome-wide pattern. To explore where in the genome we see differentiation, let's focus now on 4 locations. So let's look at the southern end and the northern end of this cluster here, the southern cluster, and then 2 of these northern locations. So 4 locations total. And what we'll be looking at the next slides are sets of pairwise comparisons between these 4 neighboring locations. So between the 2 southern locations at the bottom, between the 2 middle locations in the middle, and then the 2 northern populations. And then here we're looking at Manhattan plots of FSTs. On the y-axis, we have FST. Each dot here is a 50 kb window. And then they're organized along their position in the silverside genome with 24 chromosomes. So there's a lot going on here. If we first just ignore all the outliers and just look at sort of the central tendencies in the pattern, we see quite clearly here, just like we saw in the PCA, that across the southern part of the range, there's very little genome-wide background levels of differentiation. FST is very close to 0 throughout on average here. And then as we move further north, we see slightly more differentiation, and then the greatest differentiation in the north. So this is kind of an interesting pattern because it gives us a continuum of gene flow from high to low. So it gives us an opportunity to, at least within this system, compare what signatures of selection look like in different levels of background gene flow. So if we bring the rest of the data in here, we can also see that the patterns of outliers of what looks different from the rest of the genome-wide pattern varies across latitude. And what particularly jumps out are these big blocks of extreme islands of divergence, where we see very large blocks of 3 different chromosomes that show highly elevated differentiation compared to the rest. So I think now, as we're starting to be able to do whole genome sequencing on any organism, these kind of islands of divergence often pop up. But I think here in the silversides, they're pretty extreme in that they stretch more than 2/3 of several of these chromosomes, and the degree of differentiation is also quite extreme, where these islands here, now we're just showing averages within windows, but there are actually thousands of SNPs that are fixed for opposite alleles, with at least within these 2 blocks here on chromosome 18 and chromosome 24. So we were curious about what was causing these huge blocks of highly elevated differentiation. And to get a better look at that, Maria, my former grad student, constructed linkage maps from several different populations. So she constructed separate linkage maps from Georgia, New York population, and then from a hybrid between the 2. So basically, linkage mapping looks at levels of recombination between markers in controlled breeding crosses to infer the relative position of markers on chromosomes. And when she looked at the map she had created from Georgia, with a Georgia female, she mapped those markers against our reference genome, which was also built from an individual caught in Georgia. She found that there was quite good correspondence between the recombination distance between markers, which is showed on the y-axis here of this little plot, and the physical position in the assembled chromosome sequence here on the x-axis. It was different though when she compared the map she had generated from a New York female, where at the center of this chromosome here, that center was completely flipped around. So this is very clear evidence that there's a chromosomal inversion here, which is a well-known mutation type. And just like we would expect within an inversion, we saw that in the hybrid individual between Georgia and New York, recombination was completely suppressed. So there was basically no recombination happening along this entire block of the inversion on chromosome 24 here. So this is very clear evidence that this is an inversion that's underpinning this elevated differentiation here. And the same was true for the two other blocks of highly elevated differentiation here. Very clean pattern here on chromosome 8. On chromosome 18, it's a bit more composite, potentially, of multiple nested inversions, but the effect still being that recombination is completely suppressed in the hybrid individual. And if we look across all 24 chromosomes here, the major inversions are the ones we had already seen patterns of elevated differentiation, but there are several other inversions that popped up on different chromosomes. And altogether, segregating inversions cover, depending on how we count them, between 16 and 23% of the genome. So clearly, these polymorphic inversions play a really important role in modulating the genetic variation within this species and the variation across latitudes. And if we just zoom in a little bit more to look at how it compares to where we actually see differentiation. So here the gray dots are FST on the y-axis here, and the blue dots here are the New York linkage map overlaid, mapped to the genome here. So we can see that these blocks of elevated differentiation, sort of where we start seeing a jump from basically zero background differentiation up to this elevated differentiation, exactly coincides with the breakpoints of these inversions. So it seems quite clear that these are driving these blocks of differentiation. So the next thing we wanted to explore was how what phenotypic effects might these inversions have? So we did a QTL study where we mapped a series of traits in these controlled breeding experiments. And here we're looking at a plot of where on the y-axis we have a measure of the strength of association between a genomic region and a particular trait. And overall, we found 8 significant QTL, or quantitative trait loci, that was on 5 different chromosomes and related to 4 different traits. And what's quite interesting, so we can see that the 3 strongest signals that we have are all related to growth rate and vertebral counts. And those are found on chromosomes where we also have seen major inversions, so 11, 18, and 24. And those are the same chromosomes where we also see those blocks of elevated differentiation. This is in contrast to the other 2 chromosomes that have significant but not quite as strong associations with vertebral count and growth rate, and also one with oxygen consumption, which are not associated with any known inversion on those particular chromosomes. We don't really see elevated differentiation across latitudes at those regions. So I think the big take home from this work here is that both in the top-down approach where we're mapping traits to the genome, and the bottom-up approach where we're just looking at patterns of population of sequence variation, we arrive at the same answer, that the main signals, the strongest signals, are found on where the big inversions are. So it seems very clear that inversions play a really important role in modulating the local adaptation in this species, at least under high gene flow, because if we just think back to this pattern we saw here, we might remember that it was only in this comparison across the southern range that we really saw these big blocks popping out. if we look further north, where we see, so here each window of the genome is colored according to whether it's a putative inversion or if we think it's a centromere or telomere. And we can see that there's not the same, there's a little bit in the middle here where there's another inversion that pops out. But for the most part in the north, where there's much less gene flow, we don't see these big blocks popping out with elevated differentiation. What seems to show elevated differentiation, or what are outliers up here as likely signatures of divergent selection, is much more scattered in these small, narrow peaks across the genome here. So, of course, there's no replication here in just our single continuum of high to low gene flow, but at least what we're seeing here in the silversides seems consistent with the prediction that it is under high gene flow, local adaptation should proceed primarily through mutations of large effect or tightly linked clusters of smaller effect loci, so being these inversions, right? We don't know exactly what role they play yet, but they seem to play a much stronger role in the part of the distribution range where we have high gene flow. And it's also interesting to just think a bit more about what role do they actually play, and how strong of an effect do they actually have on these traits that we know show adaptive divergence. So here we're just looking at zooming in on the 3 largest QTL for growth rate, and then we're looking at which were associated with the inversions. And then we look at the individual growth rates for fish with the either being homozygote for the southern allele, heterozygous, or homozygous for the northern allele. And we see actually quite clear and very significant effects in all these cases. And we're estimating the proportion of the phenotypic variance explained to be more than 10% for each of these QTL. And the effect is so strong that even when we look at wild-caught fish that haven't been reared in the lab, but have just been out in the environment subject to all kinds of environmental noise, we also see significant differences in their length, which is not even controlled exactly for age, but just the length of fish that we catch in the wild. There are significant differences between individuals that carry these different haplotypes at the key inversions. So clearly, as sort of thinking of the inversions as single loci, they are pretty large effect loci that affect a trait like growth rate, which typically we would think about something being highly polygenic if we, for example, contrast to human height, which is one of the sort of the typical traits we think of as polygenic on the most recent, one of the most recent major studies that was screening millions of people. They identified more than 12,000 SNPs that account for 40% of the variance in height. So suggesting that each SNP or each variant in the human context only has a very tiny effect on body size or body length, where it's a very different kind of architecture we have here in the silverside, where we actually have these very large effect loci affecting this classically thought to be highly polygenic trait of body size. So what does that mean in practice? That's one of the questions we're curious about next. So just thinking about how these inversions are actually distributed in the wild. If we go back to thinking about, we have these locations sampled across latitudes. Here, each tiny line is an individual, where each of the SNPs within the inversion block is color-coded as to its most likely genotype, if it's a northern homozygote, a heterozygote, or southern homozygote. And then they're organized here according to their populations over here. So I think the first thing to notice is that just confirming, if we just zoom in, say we just look in this yellow location here for chromosome 24, here we can see what we were already expecting based on the linkage mapping, that also the population genomic data is showing that we basically have complete sorting with complete linkage disequilibrium across the entire length of this block, suggesting that there is no recombination between the haplotypes within populations. And then also what's very interesting is that we see when we were just comparing before Georgia and New York, we saw almost fixation for opposite alleles. But now that we're zooming in to look at the intermediate area here, we can see that there actually is a gradient of frequency from southern to northern alleles, and that there are many locations where we have both northern homozygotes, heterozygotes, and southern homozygotes. And interestingly, they appear in more or less Hardy-Weinberg proportions, so expected genotype proportions, suggesting that there's no clear evidence that there would be selection against heterozygotes or against one haplotype versus another at these intermediate locations here in the south. But we can also see that sort of the changing point from the northern to the southern haplotype becoming dominant is a little bit different for each of these major inversions here. So if we just do a plot of the haplotype frequency across different latitudes, we see that these have slightly different patterns. So suggesting that it's not a single selective driver that's governing the frequency shift on all these, but they might be driven by slightly different selection sources of selection. But coming back to this question of the genomic architecture, so if we think that these inversions are these large effect loci that make the growth rate become not a highly polygenic, but a large effect driven kind of trait, that makes sense at the kind of mid-latitude range here, where we have both northern and southern haplotypes present. But we can see that at the extremes of the distribution range, we see almost fixation, complete fixation in the south and near fixation in the north for just one of the haplotypes. So what does that mean? If we have these large effect loci that then are not variable in certain populations, does that mean that then it's highly polymorphic, that it's a polygenic trait in those populations and a more large effect trait in the other populations? Or how much flexibility is there really in the genomic architecture of this very important locally adapted trait? We're trying to address that now with some additional lab rearing experiments where we've sampled fish from different latitudes and reared them under different temperatures in the lab, and we're looking closer into trying to isolate the effects that the inversions specifically are playing, and also just looking at the genomic architecture of growth rate in terms of effect sizes in these different populations. But also circling back to this experiment I had mentioned earlier. So a few years ago, we had the opportunity to sequence the fish from this selection experiment on silverside that had shown really rapid. So the only difference, these fish here all have parents from the same location, in contrast to the photo I showed earlier. And the difference between the three groups here have evolved over just 4 generations of experimental size selection. So, what Conner and Munch did in this experiment was that they founded 6 laboratory populations, and then at the end of every generation, they would harvest 90%, either the 90% largest or the 90% smallest, or just a random harvest with respect to size. And then they let the remaining fish reproduce to form the next generation, keeping each experimental population at about 1,000 individuals. And as you could see already from the picture, here we're looking at how the weight of the total harvest and the mean weight of individual fish changed over the course of the experiment. So as we could see, from the pictures, I said, already after 4 generations, the fish where the smaller individuals had been removed were almost twice the size as individuals from the population where the large individuals had been removed. So because this was done in the lab, it was clear that it was genetically based, but it hadn't been clear what the genomic basis was. And what was really interesting here was that we found, here we're then looking at for each of these 6 experimental populations, we are comparing generation 5 after size selection back to the source pool generation 0. And then on the y-axis here, we are looking at a measure of allele frequency change. How much did allele frequencies change across different positions in the genome, organized here along the x-axis, in each of these 4 populations. And already here, one of the main conclusions from this experiment was that there was there was some degree of parallelism, but really there were different ways that the fish got smaller and larger in each of these populations. So this was already suggesting that there's some flexibility in the genomic architecture of growth in this species. And what's also really interesting is if we now, at that time, when we did this work, we didn't know about these inversions with a strong effect on the growth rate. But if we now go back and look specifically at what happened in the genomic region that harbors these inversions on chromosome 11, 18, and 24, it's interesting to see that there actually doesn't seem to be much going on the inversion 11 and the inversion 18. Only inversion 24 showed a very large allele frequency shift, but only in one of the replicates of our down-selected lines. So this is quite curious. So we already know, our expectation is we know that this chromosome 24 at least was very rare when we set up the experiment. So it seems like it wasn't present in the other down-selected line for selection to work on, which is why it only changed in one of the lines, but which highlights this very flexible genomic basis for growth rate evolution in this species. But what is perhaps a bit more curious is that we don't see any shifts, major shifts in the chromosome 11 and the chromosome 18 inversion regions. At first, I thought, oh, that must just have been because they weren't sampled in their original source pool. But now that we have the reference genome. So I forgot to say, so when we first started out doing this work, we didn't have a reference genome. So this is all just whole genome data, but mapped just to a transcriptome that we had then anchored. We had basically ordered the transcript by anchoring them to the Medaka, a related fish species. So now that we then assembled the genome and all the previous results I showed you were these original data, but now mapped to the species-specific reference genome that fortunately wasn't too different from the Medaka. There was pretty good synteny overall, at least in terms of few interchromosomal rearrangements. But it's interesting to compare just how much the patterns got cleaned up. So the top panel here is from our original results where we had mapped the whole genome reads to reference transcriptome that were anchored to the Medaka. And this is the comparison. This is showing FST between these two ends of the southern range. And we see that there's a lot of noise. There's still some clear clustering of where the high differentiation is, but there's also a lot of noise. When we don't map those same genomic reads now to our high quality species specific reference, we see that pattern cleaned up a lot more. There's still some small peaks on all the different chromosomes, but it's much less scattered than what we saw when we didn't have the really specific species reference. And then when we look at it just in windows, sort of the patterns of what pops out becomes even clearer. So based on just seeing this evolution in what we can do with a higher resolution and species-specific tool, we are now returning to that selection experiment as well and trying to take a closer look of mapping the reads of what really that rapid evolution, how that was genomically based. And now that we can easily track, we know where to look for those inversions, we can easily track the frequencies and see what happened with them in there in a much more high resolution way than we were able to before. In addition, we're also in collaboration with Søren Blikdal Hansen, who's at the Globe Institute at the University of Copenhagen. He's been doing some bisulfite sequencing. He was interested in looking into whether, in addition to the sequence level changes we saw between these groups here, if there could be an additional part of this rapid growth evolution that perhaps had more of an epigenetic basis. So he's taken a subset of the fish from these different selection lines and have done bisulfite sequencing on those. And the preliminary results actually suggest that there is a really clear signal that somehow, so here's just a preliminary result, but a bisulfite, so basically a PCA summarizing methylation patterns among these fish that's showing along the major PC axis here is showing a separation between down-selected individuals in orange and yellow and up-selected individuals in blue, the two blue shades. So it'll be interesting to see what comes of this work and to what extent we'll be able to partition sequence variants versus epigenetic, what role each of those types of variations have played in this rapid evolution and growth rate here. Thinking about expression or epigenetic and potential modulation of gene expression, it definitely seems like there's also a lot of expression level differences between these populations. So just to wrap up here, I'll just share some results from an RNA-seq experiment we did a couple of years ago, where we reared F1 offspring from parents caught in New York and Georgia under two different temperatures in the lab. And this was done by Arne Jacobs, a former postdoc. And when we did RNA-seq on those, and again, just looked at global gene expression patterns across all the genes we could get high quality data from. We're looking at PCAs here. We see that overall, the biggest, so the dots here are colored by their location, and then the open circles are the warmer temperature, the closed circles are the colder temperature. So we can see that the major axis of differentiation is separating the populations. And in this case, the hybrids cluster very close to the New York populations, which was their maternal, the maternal part of this cross. But what's interesting is that actually this separation here on PC1 is driven by more than 25% of the genes that show differential expression between the two populations. So it seems like there's a huge amount of divergence in gene expression patterns between these populations, in addition to the sequence divergence that we've talked about earlier. But what's also interesting to see is that if we look, the top plot here is PC2 against PC1. So this one here, PC2, is separating out the individuals reared at 26 versus 20 degrees in Georgia, but not really separating out the other populations by temperature, while PC3 here plotted against PC1 is separating out the individuals reared at 20 and 26 degrees in the northern and the hybrid population. So the fact that it's different PC axes that are separating out individuals at different temperatures in the two different populations is showing that it's different sets of genes that show thermal responses in the two different populations. And one thing that's super interesting is that both, so if we look at this Venn diagram of the genes that show different expression between 20 and 26 degrees, the first thing to notice here is that there's very little overlap between those set of genes that seem to be thermally responsive in Georgia and in New York, as we already saw from the PCA. But what's really interesting is that it's a much, much smaller set of genes that show differential expression between temperatures in the hybrids between those two populations. So this is potentially suggesting that these hybrids are much less, they show much less thermal plasticity, so they might be able to have much poor ability to adjust to temperature. And in addition to this, these hybrids also show substantial misexpression of genes and disruption of co-expression networks compared to what we see in Georgia and New York. So this is potentially suggesting that there could be some kind of incompatibility in these hybrids that become that are mixed between northern and southern types. And this is definitely something that we're looking into more to better understand. But it seems to contrast a little bit with what I showed you earlier about how when we go and sample in the wild, we basically see expected proportions of homozygotes, heterozygotes, and other homozygotes in each of the locations where all three haplotypes are present. So this is suggesting that there isn't really strong selection against hybrids, or at least against individuals that carry both northern and southern haplotypes at these inversions. So I think the jury is still kind of out as to whether incompatibilities play any role in maintaining local adaptation in this species here, as there are a lot of other open questions. But one thing that's clear is, I think, that inversions are important, and it's not just in silversides. So here are just a few examples from 3 other fish species that we are studying in my lab: Atlantic cod, American shad, and northern sandlands. And these are kind of Manhattan plots of FST comparisons between different populations. And you see in each case, we have these similar kind of blocks of highly elevated populations popping out that are very likely to be inversions. And of course, there are many papers being published on inversions in all kinds of other systems, especially in marine systems, but in many other systems as well right now. So I think it's a really exciting time when now we have the tools to discover that these inversions are a lot more abundant than we used to realize. And now that we have the tools to find them, we see them popping up everywhere. So the next step really is to figure out exactly through what mechanisms. They seem to play a very important role, but understanding the mechanisms through which is, I think, a very important challenge for the coming years. But in terms of the silverside, I think just to bring this back to where we started, I think the big take home here is that a species like the silverside, where if we just go out and sample in the wild, it might look like there's really nothing going on across latitudes, so across its distribution range, because phenotypically in their field conditions, all the fish just kind of look the same. But that these kind of species can actually host an enormous amount of variation. And it was only when people started doing common garden experiments that we discovered this phenotypic variation that was cryptic in the wild. And it was only when we were able to sequence their genomes that we discovered that there are these large chunks of the genome that show really vast differences across short spatial scales, that we can really start realizing the amount of intraspecific variation that probably is really important for coping with future climate change and other impacts, that we can really start seeing that and uncover this striking cryptic diversity. And this is, of course, only made possible by the immense drop in the cost of DNA sequencing. Just incredible to think about how sequencing is now more than 500,000 times cheaper than it was just at the turn of the century. And just this is old news, but just to give a little bit of perspective, thinking about the Human Genome Project completed 20 years ago, costing almost $3 billion and took 13 years for almost 3,000 researchers to complete. And now we're at a point where our little team of just a few people with a couple of years, very modest research budgets, can get a near chromosome length reference genome, can do whole genome resequencing for $20 or $30 per sample, can easily make linkage maps for multiple populations, and can map key fitness traits. So I think just this technological revolution is really democratizing whole genome analysis in a way that's super exciting, and that gives us the opportunity to start exploring much more of the cryptic variation that otherwise might go unnoticed within each of these very different types of organisms that catch our eye in the wild. So with that, I would just like to thank again all my collaborators and my lab, and I'd be happy to take any questions.
Thank you very much, Nina. This was very impressive. So many results. I don't see any raised hands just yet. So I will just start, I guess. So concerning the inversions that are not present in, like, it seems like you can't induce them when you put a specific population under different selective regimes, right? So do they capture something specific that's not in that order in like sister species, or I don't know, like how do we, yeah, what's so special about these inversions that they have an adaptive advantage?
I mean, that's really, that's, I think the very crucial next question is to understand what exactly is it? What is the mechanism through which, I think what we know now is that it seems quite clear that they play an important role and they clearly have a strong effect on some traits, but exactly how, we have no idea yet. So one of the sort of the leading theoretical expectations or one of the thoughts of why inversions might have an adaptive advantage is that it'll keep, so basically traits or alleles that work well together, right? They'll prevent them from getting separated with recombination when there's high gene flow. But because there is no recombination in the heterozygous state, it also makes it very hard to fine map within an inversion of what parts of the inversion is it that affects the trait, because they basically just get inherited as a whole block. So for example, in our QTL mapping, our QTL basically span the entire chromosome. So we're hoping, because the population sizes are so big and because we have lots of populations where we have the inversion occurring in homozygous state at very high frequencies, there should be lots of recombination in that state. And also then there should be gene conversion that over time will break down some of the recombination, that will cause a little bit of recombination. So we're hoping now with sequencing some larger population samples that we might be able to do some finer scale mapping to narrow in on where in the inversion does the phenotypic effect seem to locate to.
Mm-hmm. Okay. Ika is next.
Hello.
Hi.
Thanks enough for the talk. It was really, really interesting. I wanted to ask about, I found very interesting that the hybrids, they seem to have disrupted co-expression networks. So my first question is if you could say how you look into this disruption of the gene co-expression networks. And the second one that I found really interesting is that when you go to the wild, you seem to not see a lower frequency of hybrids as you would have expected from the transcriptomes.
Mm-hmm.
Do you have any ideas of how they, what might be going there? So I guess this suggests that in the wild things are more complicated and maybe the organisms have other ways of coping. But yeah, I wanted to hear your ideas, your hypothesis.
Yeah. So in terms of the conservation of the gene expression networks, so that was based on just doing sort of a separate expression network analysis in the two parental or the two pure types, and then comparing that to the expression network analysis in the hybrid, and then also just looking at these different kind of network preservation statistics. Yeah, I haven't looked at those details in a while. I can get back to you with details on exactly how it was done. But in terms of why we're not seeing obvious selection against hybrids, I guess one thing is that what we're seeing when we're sampling in a population where all three haplotypes are present is not necessarily hybrids between an individual from Georgia and an individual from New York, but it's offspring of individuals that are within that population and just happen to be carrying either northern or southern haplotypes at these particular inversions. So it could be something to do with that it's actually not about the inversions, this potential hybrid. So that's another thing. The first RNA-seq study we had done, we set up before we even knew about inversions. So it ended up being set up in a way where we're not able to isolate effects of inversion on gene expression patterns. So we can't really say to what extent both the difference in the degree of difference in expression patterns, and especially this disruption of networks and the higher degree of, or the lower degree of thermal plasticity in the hybrids, to what extent that's driven by inversions versus something else in the genomic background. So it could be that it's really doesn't have anything to do with the inversions, but we just don't know yet.
Cool, makes sense. I wonder, I guess it would be interesting also to sequence those haplotypes to see whether they have the inversions, but I guess it's also a question of money. But
yeah,
cool.
It's just that the way it was set up, we didn't have all 3 haplotypes represented in our different groups, since just because of what parent genotypes happen to be there. So now we actually have set up a new experiment where we were able to, from several of the locations where we have all 3 haplotypes present, we were able to breed a heterozygote with a heterozygote, so that in the offspring we'll have, within a family, we'll have both homozygotes and the heterozygotes. So then we can, in a much more controlled way, look for differences that are specifically related to the inversion type compared to the rest of the genomic background. And we'll have better power to distinguish those.
Very cool. Thank you very much.
Ricardo?
Hello, thank you for your talk. It was fascinating to learn about your research program. I was really impressed with the inversions, and I was thinking about all the debate whether the local adaptation alleles arise first and then the inversion happens to help fixating those alleles, or if we have the opposite, that the inversion happens. So I was wondering if your data allows to shed some light on this question by dating the inversion and perhaps dating the alleles inside the inversion, and knowing if it's older than divergence of the species, like we see in the sticklebacks, for example, and other beautiful systems where local adaptation is sometimes older than speciation.
Yeah, we haven't had a chance to do that yet, unfortunately, mostly because so far we've only had these low coverage data that make it a bit tricky to at least date the alleles. But what we have done is to just compare 2 sequence from 2 congeners, and we see that the amount of sequence divergence between the northern and southern haplotypes in each of the major inversions, that's all less than the genome-wide sequence divergence to the congeners. So it seems that our major inversions are of slightly different age, but they all seem younger than speciation from the extant congeners. So at least that's as far as we've gotten so far, that it seems to be it's not something introgressed from another species. It's something that at least arose within this species, but narrowing in on exactly when and dating, relative to the alleles, that's definitely something that we're interested in pursuing, but haven't been able to yet.
Thank you.
Aurora?
Hi, can you hear me? Yeah, okay. Thank you for your talk. It was really interesting to learn about your system. I'm curious about, maybe you already talk about this, but you mentioned that in intermediate locations, the proportion of heterozygotes and homozygotes are more or less according to Hardy-Weinberg equilibrium.
Mm-hmm.
And you said that it suggests that there should be no selection, or at least, this is what you read from that. But on the other side, you can see that there is a clear effect of these inversions on the phenotype, and that this should be adaptive, at least for some of them, right? So what's your way to reconcile this?
Yeah, so that's an important point, that since we know that the background genome-wide differentiation is so low, we know that there must be a lot of connectivity, and we've also seen that from other data. So the only way that we can maintain these fixed differences across latitudes would really be with a gradient in selection. So yeah, so it seems clear that there has to be selection, but probably I'm guessing the sort of the distortion of allele frequencies each generation is just not strong enough to cause genotype frequencies to diverge from Hardy-Weinberg proportions. So I was talking about at a journal club with some of you this morning, how we've been doing some temporal sampling too, where we're trying to see if we can capture exactly this dynamic, generation to generation dynamic of how gene flow might even out allele frequencies and selection then might maintain the balance. So this species is actually interesting because they have a fixed 1 year lifespan. And so they breed inshore in the spring and then grow up during the summer. And then in the fall, when coastal waters, especially in the north, starts freezing over and get cold, they move offshore. And then they come back in to spawn the next spring. So it's a nice system in the sense that gene flow happens in this one time of year. So then by sampling in the spring, we're hoping to then capture the effects of whatever gene flow has happened between one generation and the next, and then sampling those same locations in the fall, we're hoping to maybe pick up effects of selection that has then filtered those alleles that ended up in that location to match the conditions there. And then we've done that 3 years in a row to then we hope to be able to catch some of that gene flow homogenizing selection, maintaining the differentiation dynamic. But of course, the question is just how much gene flow there is, how much allele frequencies get homogenized, and if it's probably not enough to really throw genotype frequencies off from Hardy-Weinberg proportions, but maybe it's enough that we can detect with this fine scale allele frequency tracking.
Thank you.
Ethan, does your microphone work now? I'm just seeing your message now. And if it doesn't work for Ethan, I would just read it out. So Ethan wrote, thanks for the talk. Could you please explain a bit more on the link between possible genetic incompatibilities that the hybrids are having according to your RNA-seq PCA plots? Did you mean the hybrids are suffering from some sort of detrimental effects caused by the incompatible alleles they have? Thank you.
Yeah, so we don't know mechanistically very much about it yet, but just from this first pass RNA-seq study we did, we saw that there was a smaller set of genes that showed a much smaller set of genes that showed different differential expression at different temperatures that seemed to respond to being reared at a colder or warmer temperature than we saw in the purebred offspring. So, of course, we don't know how important that really is for surviving in the wild, but it just seemed like at least there's something that's being compromised. But I can't say very much about the exact function yet because the way the experiment was set up doesn't really allow us to get at that very closely. And also, we don't know how far gene flow extends in the wild. We don't know if it's mostly a matter of just exchange between close neighboring population and sort of a stepping stone type of pattern, or how much long distance migration we have, where we might actually have hybrids between Georgia and New York that could be potentially compromised. But at least in the local interbreeding, where we have different inversion genotypes, we don't observe any obvious incompatibility just in terms of who survives, at least. Does that answer?
I'm sorry, I was muted. Ethan was asking a follow-up question. Could you measure the fertility rate and so on of the hybrids and compare them somehow with the pure ones?
In principle, yes. The problem is in the lab, it's very easy to rear these fish. F1 is very easy to rear, and we take wild-caught parents and then we rear the offspring in the wild. we can also get them to breed, but it's hard to get very big batches of fish for F2. And also the fecundity is it's not clear how it gets distorted. So that's also why, of course, with the RNA-seq work, we did probably a lot of maternal and environmental effects that have influenced our results, because they were just on F1 with the parents were wild. But there are logistical constraints, unfortunately, with doing multi-generational and getting enough individual offspring to be able to set up F2. But that's definitely something that we are very interested in trying to do.
Okay, then I oversaw a question, an earlier question from the chat, from Lisa. That's the last one, then we finish. So sorry if you've addressed this and I missed it, but have you looked at how much of the variance in size is explained by all genetic components combined? Would be interesting to see how much epigenetics influence that.
Mm-hmm. Yeah, so I believe the overall heritability of the size of, or sort of the growth rate is pretty high, about 50% or something like that. I can't remember the exact number, but certainly what we capture with the inversions doesn't amount to the full amount of genetic variance explained by our data. So yeah, that's definitely something. The inversions don't explain it all. There's another genetic component that we haven't characterized yet, and yeah, and some of that could be epigenetics as well.
Okay, thank you very much. Yeah, thank you, Nina. It's been a pleasure, and I hope we will see each other again. For me, it's only left to say that please join us in 2 weeks. Natalie Feiner from Lund University will be speaking. Topic is still to be announced. Yeah, and with that,